Dear friends, good morning. Today we can discuss about enzymology, enzyme units, Metallo enzymes, metal activated enzyme, non-protein enzymes, multi-enzyme complex. First, enzyme units. Different types of enzyme units available. First one is international unit of enzyme. One international unit of enzyme is defined as the amount of enzyme which catalyzes the conversion of 1 mole of substrate into product in 1 minute under optimal condition. Optimal condition means optimum pH or temperature. Next one is ketol. Ketol is a second unit, enzyme units. It is also another unit of enzyme activity. Ketol is defined as the amount of enzyme which catalyzes the conversion of 1 mole of substrate to product in 1 second under the optimum conditions. Thus, 1 ketol is equal to 6 into 10 to the power of 7 international unit. Third enzyme unit is turnover number. The number of moles of substrate converted to the product in 1 minute by 1 mole of particular enzyme. Fourth enzyme unit is specific activity. Specific activity is a measurement of the purity of enzyme in solution. It is the amount of product formed by an enzyme in a given amount of time under given conditions per milligram of total protein. Next one is Metallo enzymes. Metallo enzymes definition: metal is tightly bound to the enzyme molecule and it is an integral part of enzyme molecule. Metals are attached to enzyme through coordinate bonds. They participate in enzyme catalysis. Example for Metallo enzymes: iron Fe2+, it is oxidases required for cytochrome catalase, xanthine oxidases, succinate dehydrogenase. These enzymes utilize are cofactor. Next one is copper. Copper, it is required for superoxide dismutase or lysyl oxidases. Third one is zinc. It is required for carbonic anhydrase, carboxypeptidase, alkaline phosphatase, alcohol dehydrogenase. These enzymes utilize the zinc as a cofactor. Next one is metal activated enzymes. Definition: in the presence of metal, some enzymes get activated, that is, their activity increases many folds. Example, chloride, it activates amylase and angiotensin converting enzyme. Second one is calcium 2+, it activates the trypsin. Next one, non-protein enzymes. Non-protein enzymes, example, coenzyme. A non-protein organic substance loosely attached to the enzyme and can be separated by dialysis and is essential for enzymatic action. It is also called as co-substrate. They serve as recyclable shuttles or groups transfer agents that transport many substrate from their point of generation to their point of utilization. The coenzyme is often derived from a vitamin, example NAD, FAD, thiamine, pyrophosphate. Next, cofactors. A cofactor is a non-protein chemical compound or metallic ion that is required for enzymatic activity as a catalyst, a substrate that increases the rate of chemical reaction. Next, move on to metal ion activators. Type of cofactors. Inorganic metal ions may be bonded through coordinate covalent bonds, example Mg2+, Mn2+, K+. Prosthetic group. These organic molecules are covalently attached to enzyme and they undergo a change during catalysis but return to the native state at the end of reaction. Next one, apoenzymes. A polypeptide or protein part of enzyme is called as apoenzyme and may be inactive in its original synthesized structure. The inactive form of enzyme present in cell is known as proenzyme or otherwise called as zymogen. The proenzyme may contain several extra amino acids in the protein which are removed and allows the final tertiary structure to be formed before it is activated as an apoenzyme. Next move on to holoenzyme. Holoenzyme means combined form of apoenzyme and also coenzyme. A complete catalytically active enzyme together with its particular group referred as holoenzyme. The protein part of such an enzyme known as apoenzyme. Holoenzyme equal to prosthetic group. Cofactor, apoenzyme, protein part. Both are combined together to form the holoenzyme. This is the active form of enzyme. Enzymes containing only protein, example pepsin, trypsin, urease, etc. Enzymes containing metal ions, example carbonic anhydrase having the zinc 2 plus as a cofactor. Arginesis having Mn2 plus ions. Next move on to multi-enzyme complex. Multi-enzyme complex means which made up of 2 or 3 group of enzymes joined together which catalyze the one particular reaction. Example, pyruvate dehydrogenase, PDH. Pyruvate dehydrogenase of bacteria and animal cells is an example of multi-enzyme complex. Pyruvate dehydrogenase enables pyruvate to enter the TCA cycle by catalyzing its overall conversion of acetyl-CoA. Overall reaction pyruvate plus CoASH plus NAD which gives the acetyl-CoA plus NADH+. The pyruvate dehydrogenase complex is a multi-enzyme complex, multi-molecular aggregate of enzymes which consist of 3 enzymes: pyruvate decarboxylase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. These 3 enzymes combine together which catalyzes the pyruvate to acetyl-CoA formation, generally named as pyruvate dehydrogenase. Enzyme is located in the mitochondrial matrix and it contains 5 different coenzymes like thymine pyrophosphate, TPP, lipoic acid, coenzyme A, FAD+, and NAD+. The reaction catalyzed by pyruvate dehydrogenase complex involve the following steps. First one, pyruvate is first decarboxylated to form a hydroxyethyl derivative of thymine pyrophosphate, the coenzyme of pyruvate decarboxylase. Second step is the hydroxyethyl derivative then reacts with oxidized lipoic acid, a coenzyme of dihydrolipoyl transacetylase. This results in acetyl group which bound as thioester to side chain of lipoic acid. Now the acetyl lipoic acid reacts with coenzyme A to form acetyl-CoA and reduced lipoic acid. This reaction is catalyzed by enzyme dihydrolipoyl transacetylase. Third one is the reduced form of lipoic acid is oxidized by FAD dependent dihydrolipoyl dehydrogenase enzyme, leading to regeneration of oxidized lipoic acid. The FADH2 is reoxidized to form FAD plus by dihydrolipoyl dehydrogenase in the presence of NAD plus. Now the acetyl-CoA is the major product to enter the TCA cycle. This is the overall reaction of pyruvate dehydrogenase. The pyruvate in the presence of thymine pyrophosphate, carbon dioxide liberated to form the acetyl pyrophosphate. Then the acetyl thymine pyrophosphate reacts with lipoate to form the acetyl lipoate. The acetyl lipoate then reacts with the coenzyme A to form the reduced form of lipoid which releases the acetyl-CoA. The reduced form of lipoid then reacts with FAD to form the FADH2 and then release the oxidized form of lipoid. The reduced form of FADH2 then reacts with NAD to form the FAD. Finally, we can get the reduced form of NADH H+. Here we are producing, here we can get the acetyl-CoA. Next one example, fatty acid synthase complex. This is also multi-enzyme complex. Fatty acid synthase complex is a dimer. Each monomer is identical, consisting of one remarkable polypeptide chain containing all 7 enzyme activities and one ACP with a 4-phosphopentathenic group. In close proximity is another thiol of cysteine residue of 3 ketoacyl synthase of other monomers. This is what about because the two monomers lie in a head-to-tail configuration. Since both thiols participate in the synthase activity, only the dimer is active. Each subunit consists of ketoacyl synthase, acetyl transacetylase, malonyl transacetylase, hydratase, enoyl reductase, ketoacyl reductase, acyl carrier protein. Eighth one is thioesterase. This is the diagrammatic form of fatty acid synthase enzyme complex. These two monomers join together to form the dimer. The function of fatty acid synthase complex is homodimeric, which consists of 552 kilodalton molecular weight enzyme that regulates the de novo biosynthesis of long chain fatty acids. This cytosolic enzyme catalyzes the formation of 16-carbonatum palmitate from the acetyl-CoA and malonyl-CoA in the presence of NADH H+. Reduced form of NAD will be required for fatty acid synthesis. Thank you.